Okay, so it's 12 o'clock. It's my pleasure to welcome you all to today's seminar in the DF HCC Connect Science seminar series. I'll remind you that to please go to the website so you can see recordings of previous talks and also the upcoming schedule. And for today's talk, also please post your questions in the Q&A tab and not in the chat box. It's my real pleasure to introduce Tom Look, who is from Michigan. He went to college, medical school, and did his pediatrics residency there. And then he went to St. Jude for a pediatric hem-onc fellowship. At St. Jude, he worked as a postdoc with Chuck Schur, and then launched his independent lab there. He rose rapidly through the ranks. And then we were lucky enough in 1999 to recruit him here to Dana-Farber, where he's now professor of pediatrics. Tom has made really a remarkable number of seminal discoveries. As a fellow, I think he discovered the prognostic value of DNA contact in leukemia and in neuroblastoma, and also the prognostic value of MYC amplification in neuroblastoma. And these biomarkers are still used today, 30 years later, in patient treatment decisions. He then went on to clone several of the major oncogenic transcription factor translocations in leukemia, and his work has shed fundamental insights into the mechanisms of oncogenic transcription in principally in leukemia, but also in neuroblastoma and other tumor types. And more recently, he's focused his attention on PP2A, which brings back some good memories for me. So Tom, it's a pleasure. Look forward to your talk.
Great. Thanks so much, Alejandro, for the great introduction. So as you'll see, it's very fitting that Alejandro is the moderator because he actually initiated this project in my lab in 2007. So today I'll talk to you about molecular glues that are able to activate specific flavors of PP2A tumor suppressors, meaning ones that contain different regulatory subunits that target them to dephosphorylate different targets in the cell. And I'd also like to thank Nellie Pollack and Dave Weinstock for organizing this series, which I've heard from my colleagues is really popular around the country and the world. So thanks to them. Okay, just a second here. There we go. So the work I'll present today is primarily performed by Ken Morita, shown here, who arrived in my lab 3 years ago. And he's published one major paper on this work already in 2020, as noted here. His efforts were in a close collaboration with Nathaniel Gray and members of his lab, and Eric Fisher and members of his lab, making a powerful team combining structural biology and medicinal chemistry with leukemia cell biology. I have disclosures here for Genju Therapeutics and Omega Therapeutics. And here we go, Alejandro. I wanted to give you the history of this project because I think it makes it more interesting if you hear the story. And this project started as Alejandro's first project as a pediatric oncology fellow when he joined my lab in 2007. And so he initiated a repurposing screen, screening known FDA-approved compounds for their ability to kill MYC overexpressing thymocytes. And the schema is shown here. Alejandro is shown over here in a more casual mode, pursuing striped bass on Boston Harbor. And you'll notice striped bass really look very similar to zebrafish, only of course the zebrafish are only about 1.5 inches long at maturity. So with this screen then, he managed to screen 2,500 FDA-approved drugs, and he had to set up a really new system in my lab to make MYC inducible with tamoxifen. in order to be able to separate the fish in time to add the drug and then treat both MYC expressing, which you could tell by the DS red, which MYC expressing, which he could tell by a marker, a pigment marker in these fish, and ones that did not overexpress MYC. And of course, the active drugs then reduced the DS red fluorescence in the thymus of these fish. So to make a long story short, his major finding was that a group of phenothiazines was able to potently shrink thymocytes in the fish. And this is obviously a new activity for these drugs, which are primarily thought of as major tranquilizers and block dopamine signaling to help with schizophrenia. So the best drug of this group was perfenazine. And so then Alejandro faced the problem. How was it working? So obviously it could be working through the dopamine receptor, which is one of its known activities, but it has other activities also against protein kinase C, calmodulin, integrins. And so this became a real problem. In order to publish it, we really needed to know what the pathway was that it was acting through. So at this point, Alex Kenseth stepped in. He was another talented ped hemoc fellow, actually recruited to my lab by Alejandro, who was expert in mass spectrometry, is expert. He's now at Memorial Sloan Kettering Cancer Center. And he was able, using an ingenious approach of actually competing binding proteins off of a perfenazine pull down, using different compounds in the order of their activity. And he looked for a binder to the perfenazine pull down that competed off with the same kinetics as the activity of the drugs in killing thymocytes, or TALL, let's say. And so, and this actually worked, and he found that the target is the structural subunit, PP2A, 21RA of PP2A, and that it actually worked by activating the enzymatic activity of PP2A. So that's shown here. And this resulted in a paper. So 7 years after he started this project, Alejandro did obtain a nice publication on this finding, along with John Astor's lab. We joined forces with John Astor's lab sort of in the middle of this project to actually finish it off. And so this, and then Alejandro, of course, I think by this time he was already running his own lab at Children's. So it just shows you how long these things can take. And so now I think I should give you a little background into PP2A, because this is not an easy area. So PP2A is actually 60 different phosphatases. It's a heterotrimeric protein. The structural A subunit and catalytic subunits are pretty easy. There are only 2 of each of these. And the A version is primarily used in most settings in cancer cells. But the B subunits are much more diverse. And so there are several categories of B subunit shown here. They have these long PPP names, but you can refer to them just as the protein names. B55, alpha, beta, gamma, delta, then B56, and the so-called, and this group, and then the striatins, which I'll get to in a minute. And so, as I said, there are 60 possible heterotrimeric enzymes, And together, these different forms of PP2A account for about 70% of dephosphorylation events that occur in cell physiology. So of course, PP2A is a well-known tumor suppressor. And interestingly, each of the 19 genes encoding the subunits are rarely mutated in human cancers. So it's different from many tumor suppressors. And it's kind of cool because the subunits are intact and the RNAs are expressed and the proteins are expressed. So it can be activated, where in most cases, if the tumor suppressor is inactivated by mutation, it's very hard to reactivate it in cancer cells. So how does it work? Well, actually, the heterotrimeric assembly of PP2A is opposed by a whole set of inhibitors in the cell. And so by upregulating the inhibitors, one can inhibit a given enzyme, and by the levels going down, then it can be activated. It's kind of, I'm not sure this is how it really works totally physiologically, but at least it's an important aspect in terms of cancer. So as you'll see, PME1 is a prime inhibitor that's overexpressed in most human cancers. And so now with this background then, we're all excited to activate PP2A to suppress actively growing human tumors. But there's a major problem. And that is, of course, that although it activates PP2A, it also, it cannot be used. Profenazine cannot be used because it inhibits dopamine signaling at concentrations lower than necessary to kill TLL, or you'll see other cancer cells. And so this inhibition of the dopamine receptor signaling is not needed for anti-cancer activity, but it causes serious complications in patients, and it just precludes using profenazine or really any of these compounds as anti-cancer agents. So this is when Ken Morita joined my lab, I think about 3 years ago. And so his project was to select for a small molecule related to profenazine that would have lost this ability to inhibit dopamine signaling and would accentuate the ability to activate PP2A. And so in this way, get around this toxicity and emphasize the anti-cancer activity of this family of small molecules. So to do this, he used 3 assays, a typical IC50 assay. As you can see, profenazine has an IC50 of around 5 micromolar. And biochemical assays for PP2A activity. And this cute little assay measures the release of free phosphate from a universal substrate that's added, this peptide. And then with malachite green absorbance, one can measure the amount of free phosphate emitted and determine the activity of any different compound in activating PP2A. And then finally, for the dopamine receptor signaling, he set up an assay in HECT cells that involved introducing the dopamine receptor, GQ5I, SRF, and a reporter. So the way this works is that if you add dopamine, it inhibits the dopamine receptor, and one emits luciferin. You see bioluminescence. And so, of course, when you add profenazine, it will block this signaling, and you'll no longer see luminescence from the luciferase. So this is the assay then to find drugs that don't inhibit dopamine receptor signaling. So the end result, he tested 81 compounds related to perfenazine, and this graph has all three assays results on it. So if you just look at perfenazine, you can see on the x-axis, the IC50 is here around 5 micromolar. And then it has, we set its phosphatase activity at 1.0. And then you can see it's rather a large green circle. This means it has potent inhibition of dopamine receptor signaling. So with these properties in mind, obviously the one we're most interested in here from this screen is IHAP1. So this compound is a very little bubble on the plot, meaning that it has very low ability, not even any measurable ability, to inhibit dopamine receptor signaling. It has more PP2A activity than perfenazine. It's about 10 times more potent than perfenazine, about 500 nanomolar instead of 5 micromolar. And so this became our go-to compound to activate PP2A through this group of small molecules. So what about in vivo? Well, as many of you know, my lab works in fish, and Chuning, he is a leader of this work in my lab. And she did these studies to have a bioassay to reproduce this effect on dopamine receptor signaling. So what she found is, first of all, if we overexpress MYC to induce TLL in the fish, it induces a tumor here that expresses GFP, so it's easy to see. And the fish with large TLL tumors can be treated with perfenazine or IHAP, and indeed IHAP is more active in shrinking the tumor cells. But that's not all. The fish also depend on dopamine signaling for their ability to remain upright and swim in the water column. And so if you treat the fish with perfenazine, they almost immediately flip over and kind of wiggle on their back. And so that's the sign that they've lost motor control from the basal ganglion. They can no longer maintain their upright swimming posture. And of course, with IHAP1, we observed that, or Chuning observed that the fish have no problem with dopamine signaling. So what about anti-tumor activity in mice? Obviously, sooner or later, we have to move from fish to mice. And so this experiment by Ken Morita is shown here, where the mice were given a TLL cell line that we use, COPDK1, as a prime example of a TLL line that's very responsive to IHAP1. And then a series of mice treated either with vehicle or perfenazine or IHAP at 2 different concentrations. And the second concentration here is 80 mg per kg per day orally. And that's the maximum that is soluble to give to the to gavage the mouse with every day. And so the results are shown here. You can see what happened is that the mice, even at 2.5 mg per kg, so below the IC50 for perfenazine down here, the blue line, there was no increase in survival over DMSO. And we know if this is pushed, you can make the mice respond to perfenazine, but at those doses, they're already basically statues. They can't move because of inhibition of dopamine signaling. By contrast, the IHAP1 at both doses prolonged the lifespan of the mice very nicely, and was very active in the beginning, and we think probably failed because of pharmacokinetic properties, because it apparently induces its own destruction in the liver after 10 days or so. So just looking at the bone marrow of these mice, the other interesting finding was that if you look at the DMSO control with H&E, it's at this point, this is at the time that the normal mice are ready to die. They're of course packed with leukemia, and the IHAP treated mice have completely normal bone marrows. So the leukemia cells actually shrank over the 7-day treatment period. Well, and here's the anti-human stain, just showing these are really human leukemia cells in this xenograft system. And over here, you see that there aren't any detectable leukemia cells, pretty much. Although we know that they're there because they do grow back over the next 2 weeks. So this looks kind of cool. We have IAP1 killing the tumor cells through activation of PP2A in a way that strangely is not toxic to the proliferating cells in the mouse, the bone marrow in this case, and the blood counts were completely normal after prolonged treatment with IHAP1. So the question is, what is the substrate of PP2A that forms when you treat cells with IF1 or profenazine? And so Ken performed a phosphoproteomic SILAC experiment, which to my surprise revealed the substrate. And you can see here in red, the MIB-L2, both with perfenazine and IHAP in cells, repeated 3 different independent times and so forth. One substrate was consistently dephosphorylated in the cells treated with the drug compared to control. So this shows a little bit about the phosphoproteomic SILAC approach. So that's MIB-L2. Well, what is MIB-L2? MIB-L2 is also called BMIB, and it is a relative of the more, I'd say, famous MIB, C-MIB, that is known to was an oncogene found in early days as an oncogene that caused leukemia. And you see the structure of this protein. It has an activation domain, and it has a MIB-related DNA binding domain. And this actual gene is the primordial ortholog of Drosophila MIB. So it's the most closely related to Drosophila MIB, and it has the same function as Drosophila MIB in that it is a master regulator of essential genes encoding cell cycle proteins. And if you delete this TAD, it's already known you disable the transcriptional activity of MIB. So it was nice to speculate that maybe this serine 241 that we found dephosphorylated was actually required for transactivation activity. Well, of course, then we began reading about BMIB, and I think the best review we found is from Jim DiCaprio at Dana-Farber, who's worked on this for years. And the essential aspects are shown here. It's really a fascinating complex. The DREAM complex is the complex that represses cell cycle genes during G1 phase. So obviously the cell doesn't need to express all these genes required for S and G2 and M phase. So they're all repressed by one complex. And this DREAM complex has E2F4 and P130, an RB-related protein, and a complex called MUVB, which actually is 5 different transcription factors. And so basically, these control the DNA binding. And in this case, P130 is responsible for repression of the genes in G1. Well, as the cells move into S phase, then the RB and E2F4 are lost, and BMIB becomes expressed and joins with MUVB. And this is called the MMB complex. So BMIB itself is very cell cycle specific. It starts expression at G1 and then it's degraded in M phase. And as part of this complex, then it regulates at least 200 genes that are required for S and G2 phase. And so armed with this knowledge, then, we look carefully at the cells treated with perfenazine and IF1, and we can find the same finding, which is rest of the cells in 4N phase. This would be in these DNA histograms, 2N, 4N. And you can see the 2N cells go down relative to the same total number of cells, and the 4N peak rises. S also rises. And so then we see the same thing with IF1 and the same thing if you knock down BMIB. So this seems to be really a cellular marker of the activity of BMIB in the cell. And you can, using antitubulin stains, you can see what's actually going on. Here shows a normal mitosis in cells treated with DMSO, and you see the bipolar spindle. So the centrioles migrate to opposite poles, and with the microtubules that form, pull the chromosomes apart that are all at the plate. Well, in treated cells, we don't see that. We see just a flare out of the tubulin, microtubules, shooting all out from the center of the nucleus. And this is called monopolarity. So obviously, this was my first exposure to monopolarity, and we went to the work of Tim Mitchison, who's worked on this extensively. And he has really beautiful immunofluorescence photomicrographs of cells showing a normal metaphase here. So here you have the two centrioles in the opposite poles of the cell. In the middle, this faint blue are all the condensed chromosomes lined up along the spindle and being pulled apart so that you can generate two identical daughter cells. And of course, when you don't have MIB-L2, you don't make the proteins that are required for these centrioles to migrate to the opposite pole. So they stay in the middle. And this flare of microtubules out from the middle is caused by the lack of movement of the centrioles. So this shows you see the same thing with MIB-L2 knockdown, same exact phenotype. And so then we went to the work of Ian Cheeseman. So over at the Whitehead. So his lab had done this screen in HeLa cells of knocking down candidate genes, a sort of a candidate gene screen for genes involved in mitosis, and divided them up according to the phenotype of the cells. And this category here in yellow, he calls spindle bipolarity. That means they don't have bipolarity. So that's our same phenotype, monopolarity. And he found that knockdown of each of these genes is sufficient to produce this phenotype in the cells. So each of these proteins is required for a normal bipolar mitosis to occur. And if you lack any of them, you get this monopolarity, or as he called it, spindle bipolarity. So Ken took a look at these genes, and in our cells, when you knock down MIB-L2, the levels go down, as shown with two different shRNAs shown in green. And then the drug, each of the drugs does exactly the same thing. So we feel confident that the mechanism here is mediated through the dephosphorylation of MIB-L2 S241, leading to inactive MIB in terms of transactivation and the inadequate expression of these protein targets of the MIB-L2 MUVB complex. So that was kind of, to me, amazing. And what we think is going on is actually the PP2A that we're making probably dephosphorylates multiple different substrates, but most of them, the kinase can compensate. And so the cell doesn't actually die. It may go through a period of compensation. A kinase is upregulated to account for the phosphatase being activated, and the cells proceed. But apparently in this case, this circuit is disconnected, and the kinase that puts on this phosphate no longer can replace it when you introduce and activate the phosphatase during the cell cycle. So I found out, I hadn't worked a lot with kinases before, but obviously the big advantage is one can actually prove things about how they're working. So Ken put in the S241A and S241D mutants and wild type after knocking down MIB-L2. So what you see is that the wild type, which is here, the third row, here's what happens when you knock it down, 4N. Wild type rescues. The S241A does not rescue. So this phosphorylation is required for activity of MIB-L2. And the S241D phosphomimetic version of S241 is perfectly fine, and not with MIB-L2 knockdown. And it's also perfectly fine when you treat with IHAP1. So you can see this little bar graph shows that the cells don't have normal viability when you treat, when they have this phosphomimetic version of MIB-L2, even though you're treating them with IHAP1 or perfenazine that normally reduce their growth by 50%. So that was kind of fun. So we now shown that this phosphorylation S241 on MIB-L2 is required for MIB-L2 to transactivate the products needed in prometaphase for bipolarity. And the sensitive tumor cells are arrested with monopolarity. And interestingly, normal bone marrow cells are not nearly as sensitive to the drug. And we're still working on the mechanism, but they have a means of bypassing this strict requirement for MIB-L2 to get through mitosis. So now we came to the question of which PP2A is activated. So I already told you there are 60 different enzymes, 16 different B subunits, and obviously you'd like to know which PP2A you're working with. Calling them all PP2A is like if you called all tyrosine kinases SARC, and there weren't names for each of them. So it's really essential that you know which substance of matter you're dealing with here in this field that's kind of confusingly just lumped together as PP2A. So you can see all the different B subunits. That's the key part. We've clearly, and the A and C mostly use A alpha and C alpha, and A beta and C beta seem to be somewhat interchangeable. So we're not sure that there's too much specificity in A and C, but the B subunit is critical. So for this, Ken did a focused CRISPR screen and looked at just the IC50 for starters, and he found that the PP2A that's activated by perfenazine or IHAP1 is composed of this subunit A of A, the A subunit of the catalytic, and this one, which is B56E. That's an easier way to remember it than this complicated name. So it's one of the specific B subunits is formed, and it's not a general activation of all PP2As, but this one specific trimeric complex. So he was able to show this also by co-immunoprecipitation. So if you put all the subunits in either in cells or in insect proteins, they don't come together unless you add the drug. And then you get the complex so you can pull down the A and this version of E with an antibody. And you can blot it after you pull it down with anti-C. So that now we know which PP2A is activated. Now at this point, I want to go through other work that's going on simultaneously in Gautam Narla's lab. And he's been studying over about the same period we've been studying PP2A, a compound called SMAP that they found, and derivatives of SMAP. And so this compound has this structure here. It has a heterotricyclic ring and some other aspects. They're sort of similar to perfenazine and IHAP1, but it's obviously different in some ways, and it has different properties. So that leaves the question then, do our compound, and that's shown here, you can easily compare the structures. So the question is, do our compounds activate the same PP2A as SMAP? And so Ken had his, and first of all, in the phenotype, Ken found that while I have block cells in 4N in mitosis, clearly SMAP block cells in G0, G1. And Gautam Narla's found the same thing. So even on a cellular phenotype basis, it looks like they're quite different. And Ken this shows his experiments. He did them all the different ways. But if you do IP Western, so it's a Western for each subunit of complexes immunoprecipitated with anti-catalytic subunit, you see that for, as I told you already, for perfenazine and IHAP, we get this complex that contains the PPP2R5E subunit, and really none of the rest of them come down here. This one has some background in the STRN. And then if you treat with SMAP, you get a totally different B subunit, the same catalytic and structural subunit, but a totally different regulatory subunit. In this case, this one, which we call B55 alpha. It's a lot easier to say than this long name. And so here we are. You can see the idea then that our compounds make this enzyme, and SMAP makes a different enzyme that has different tumor suppressive properties in block cells in progression from G1 phase into S phase. So I have some diagrams. Now, Ken has taken a look at the structures of these molecules, what is known in the literature, and tried to draw representative structures. And you see now in cells, we know that C doesn't exist by itself. It always heterodimerizes with A. So one can envision a CA heterodimer. And then it's been shown that the C subunit then can be methylated on the tail of the catalytic subunit by a methylase called LCMT1. And this is a critical step because as soon as the C subunit is methylated, then it spontaneously forms a complex with each of the B56 subunits, that family of B subunits, as shown here. And this heterotrimer then is fully active as a PP2A phosphatase. And this apparently is at least part of what's going on in cells when the cell triggers the formation of the phosphatase for a physiologically needed dephosphorylation, often in the cell cycle or other aspects in different systems. So that's how it normally seems to occur. Now, what's the story in cancer cells? Well, in cancer cells, we start at the same place. However, cancer cells overexpress very high amounts of PPME1, and this is a direct inhibitor of LCTM1. So it demethylates the exact same methylation site on the tail of the C subunit. So that renders this CA unable to complex with the B56 family I just showed you, and rather it spontaneously then forms complexes with this group of STRN B types of B subunits, which are really quite different in structure. And they form a complex called STREPAC. And this complex paradoxically promotes oncogenic transformation. So the cancer cells overexpress PPME1 to specifically make this version of PP2A, which then promotes transformation. And Bill Hahn has been one of the major investigators working on the STREPAC types of PP2A complexes. And here you can see in a paper from Jim DiCaprio, it seems to act by dephosphorylating a MAP kinase, a version of MAP kinase, and acts through the YAP pathway to promote tumorigenesis. So we have cancer cells then, and virtually all kinds of human cancer have this PP2A subunit and high levels of PPME1, so they really can't make the B form of PP2A. So now we come to the molecular glues. You notice I mentioned molecular glues in my title. And this is a field that was recently reviewed by Stuart Schreiber in Cell 2021, 1 month ago. So if any of you are interested, you can refer to this review, which is excellent. And Stuart reviews the discovery of the first molecular glues, which are natural products, cyclosporin, FK506, and rapamycin. And you can see here that they are immunosuppressants acting through cellular proteins, calcineurin for the first two, and mTOR for, of course, for rapamycin. And these act to prevent activation of T cell signaling and growth in response to transplant, for example. So they repress rejection of transplants. Okay, then a little further down the line in this molecular glue area, chemical inducers of proximity were discovered. So these are not natural products, they're simple synthetic therapeutic compounds that are specifically act to stimulate endogenous pathways through their ability to induce proximity of two proteins. So of course the prime examples are lenalidomide and thalidomide, which are now known to bind to cereblon and mediate ubiquitination of discrete target proteins. And of course the targets were discovered at Dana-Farber as Ikaros proteins for in multiple myeloma by Bill Kalin, and CDK1A in MDS by Ben Ebert several years ago now. So this field then has progressed to the development of PROTACs, which I think everybody knows is designer bifunctional molecules that are all the rage now to specifically degrade proteins in cells through, once one has a binding moiety that one can synthetically hook onto the degradation system. So this field then is obviously a very fast-growing field, and this area we're studying is a part of this field. So we're also studying small molecules that act as molecular glues, so synthetic molecules. In our case, perfenazine and IHAP1, in Gautam Narla's case, SMAP. So in this case, we're starting with a cancer cell. So it has many of these complexes, the street-pack oncogenic phosphatase. And then we add perfenazine, and it acts as a molecular glue to bind to the CA. We're not sure exactly how, but somewhere in the CA. And presumably, when it binds, creates an altered conformation. And so this STRN protein is expelled from the AC heterodimer, and it goes on now to acquire the property of being able to form the classic B56 types of PP2A, such as the one we're studying, B56E, even though the catalytic subunit remains unmethylated. So even though it's unmethylated and normally it wouldn't make complexes with the B, now the allosteric structure has changed in such a way that it attracts B56E specifically. So I'm getting close to my time. I have a few more minutes, and just to go through some of the sort of cancer biology of our compound, IHAP1, this series. And so obviously many of you might want to know if it's active against only TALO. So the answer is it seems to be quite generally active against human cancer cell lines, at least, where we're able to test it. So this actually shows the depth map analysis for CRISPR and activation of MIV-L2 from the Broad, where they have over 700 cell lines, all treated with 5 different CRISPRs and a dropout screen to determine which genes, when inactivated, cause the death and lack of growth of human cancer cell lines. So the most sensitive lines are on the top of this chart. So colorectal is actually the winner, the most responsive to loss of MIV-L2 in terms of cell death. Leukemia is second, so we're studying a very sensitive tumor. But you can see actually this level here, -1, is a very severe dependency in this screen. And even -0.5 is generally quite remarkable in terms of the loss of ability of cells with inhibition, in this case, inactivation of MIV-L2 to grow. So actually, most kinds of cancer shown here are quite sensitive. And that includes neuroblastoma with high expression of MYC-N. And we decided to choose one other example, lung cancer, quite active in many different cell lines from non-small cell lung cancer. So the interpretation then is that MIV-L2 is a relatively non-selective and pretty strong tumor cell dependency, which is often something you don't look for in the screen because you'd be worried that it'd be toxic to normal cells. but we've already learned that our drug is not toxic. And as I said, we're still trying to figure out exactly why, but normal cells seem to be able to carry on with proliferation, at least at a level required for normal physiology, despite the presence of these drugs. So just to show you, of course, in neuroblastoma then, we can test these cell lines then with the drugs to double check the findings genetically. And we see that neuroblastoma cell lines are responsible. They undergo caspase-3 cleavage, indicating cell death when treated with adequate amounts of these drugs. Lung cancer lines are quite sensitive. And interestingly, we seem to see the same finding that showed up in DepMap, which is MIB-L1, a relative of MIB-L2, can substitute. So if MIB-L1 is very high in lung cancers, they tend to be more resistant. And if MIB-L1 is very low, they tend to be more sensitive to, in this case, IHAP1. So then the question that we're working on is, why is dephosphorylation of MIB-L2 on this in this serine residue not toxic to normal, for example, highly proliferative bone marrow cells? And I'm going to leave this as a question for the future. That's one we're actively working on now. And we have some data on synergy of our molecule with other drugs. I'll just close by mentioning that Ken is working on ways to try to find other kinds of small molecules that activate different PP2As that may have different activities in terms of specifically interfering with diseases of different types. PP2A is supposed to be involved in everything from heart disease to rheumatoid arthritis to neurodegenerative diseases to every kind of cancer. So there's really maybe quite a potential here to find additional molecular glues that can activate PP2A in other contexts. And finally, I wanted to mention that one of the most exciting things being done by Eric Fisher's lab, who's been able to crystallize PP2A in complexes induced by perfenazine. So, and it crystallizes as 2 units in the minimal structure shown here. And Ken is now working with Eric and members of his lab to try and determine exactly where the compound is in this structure so that this can be used to more precisely improve the molecule by medicinal chemistry. So I think I've mentioned everyone going through the talk. I'll just again thank all the people who worked on this project, and thank you for your attention.
Great, Tom, that was terrific. I'll remind everybody again to please enter your questions on the Q&A tab. We have several already. Some of these actually came before the last part of the talk. So Juco Westermark asked first about the lineage specificity, which you addressed later. But he also is wondering about the specificity towards MIB-L2. This is quite astonishing, thinking of the various potential targets of PP2A. Any idea why this could be so MIB-L2 specific?
Well, you know, the basic idea is that this specificity is specific for B56E, the B subunit. And the reason it's specific is because perfenazine only makes this kind of PP2A and not the others. And even the phosphorylation or the methylation of the catalytic subunit, when Ken did that in cells, it would form all the different B subunits. So this molecular glue is more specific than some of the inhibitors in terms of which complex it stimulates. And presumably that has to do with aspects of how it influences the structure of AC and just which B subunit is attracted in then to make the heterotrimeric complex. And those are the details that we're really not going to know until Eric manages to, he has the structure, but we can't so far find the perfenazine in the structure. So there are many tricks that he's using to find this out. And when we do, presumably we'll know more about the basis for the specificity.
A couple of questions about the potential toxicity. One is, how long can fish tolerate IHAP1 treatment? And the second one is whether MIB-L2 depletion is lethal for normal cells.
Right. So I think at some level, MIB-L2 depletion is toxic to normal cells, but not in the ranges we're inducing by IHAP and the other drugs. So in the mice, at least, we have a solubility problem with IHAP1. And so we could only give so much of the drug, 80 mg per kg. But at that relatively high dose, we saw absolutely no toxicity of any kind, no organ problems, no bone marrow problems. Blood counts were normal. The blood counts recovered while they were receiving the drug. And the platelet counts actually increased. And that may be related to megakaryocyte polyploidy. We looked at that a little bit. But it's possible that actually it makes the megakaryocytes more polyploid and actually increases the platelet count. So the answer, the long answer is we don't know exactly why it's not toxic, and we're trying to work on that. It apparently has to do with checkpoints that are lost in the cell cycle when cells become malignant. And so normal cells are able to slow down the cell cycle and wait until these proteins are made in adequate amounts before proceeding on. For example, into mitosis, where they can't succeed if they don't have the proteins they need for the centriole separation.
All right, great. Another question, does the SMAP activated PP2A complex dephosphorylate MIB-L2?
No. So you'll notice I didn't go into too much about that. And actually, the SILAC phosphoproteomics Ken's done multiple times, and there's no consistent single protein like he found for MIB-L2. And I think really it's quite fortunate that he was able to find MIB-L2. Turns out the TLL cells express very high amounts of MIB-L2 compared even to other cancer cell lines. And so it's a perfect system to see that substrate. There are reports that SMAP actually stimulates a dephosphorylation of MYC, leading to loss of stability of MYC. And we're trying to confirm that, but we haven't. That's Gautam Narla's work, and we don't have any data on that. All right, it's a feeling. It might work through MYC since you see a G1S block.
Oh, great. Maybe I'll jump in with final question. You know, since this is conserved so well from fish to humans, it seems like it's under positive selection. Do you think there's a physiologic ligand for this site, maybe an endogenous metabolite?
Right. So we've been thinking a lot about that. This is, I think, attractive because to me, in normal physiology of all the different cells in the body, it seems like kind of a blunt way to control PP2A to just have inhibitor levels going up and down, regulating this. So it's attractive to think maybe there are chemical mediators in cells that perform this kind of activity, but we haven't found any yet. So it's still kind of an open question.
All right, we have one more minute. So last question here. The question is whether the molecular glue will work in tumors that have PP2A mutations of PP2A subunits.
Okay, obviously the answer there is no, if they mutate the specific ones. There are some tumors that mutate the A subunit. There's a paper from Gotham's group on literally hundreds of tumors that have been sequenced, as you can imagine. And the percentages are really low. The A subunit is only mutated in 1% of human cancers. But there's a group, I think, uterine, that has slightly more than the others. So there may be something going on. We don't see mutation of B56E, the one we're studying, or B55 alpha, really in any human tumor cell lines. So it really does seem that these proteins are there, they're expressed, they're intact, and they're ready to be reactivated given the right small molecule glue to do it. It's a perfect system for reawakening tumor suppressors in cancer cells.
All right, great. Well, thanks a lot, Tom. That was a terrific talk. And thanks everybody for joining.